Hello guys, welcome to Agriclarity, a sister channel of Clarity for Sure. And I welcome you all to yet another episode of Concept Clarity Lecture Series. This is the second part of Pomology. In the first part of Pomology, we've discussed about the different fruit types and the classification of fruit from the examination point of view. During this session and also at the end of the session, I've asked you certain questions. So before starting today's session, let us first see the answers for those questions. So while discussing about Sorosis, I asked you to give another example of the fruit type Sorosis apart from jackfruit. So the answer for that question is pineapple. Sorosis is a type of composite fruit. At the end of the session, I asked you another two questions. That was, what is the fruit type of bear? Whose answer is Drew. And the next question was, which family bears pome fruits? So the answer to that question is Rosaceae, which is the apple family. Today we are going to discuss about tropical fruits, and in today's session we'll discuss about the three most important tropical fruits, that is a mango, banana, and papaya. So in this session we're going to discuss about those most important, most vital points related to the three crops which have 100% probability of appearing in the exams. So we know that the secret to success in these competitive examinations is to filter out the data which is important and which is not, right? So therefore, please pay attention to everything I say during the session, because there are certain points that are not being written here, but that I'm going to speak. So please pay attention. And once you revise this session 2-3 times, you will have covered almost 90% of whatever important data are there related to these three crops. So let us start one by one. First, we'll start from mango. So botanical name of mango is Mangifera indica, and family is Anacardiaceae. You know that even cashew nut belongs to the family of Anacardiaceae. Chromosome number is 40, and a typical example of amphidiploid fruit is mango. So in the exam question, maybe give an example of amphidiploid fruit. So then you'll have to write mango. The cross-pollination is in mango is done mainly by housefly. And in the previous session, that is the first part of pomology, we've discussed about drupe fruits, right? And there prominently I've given the example of mango, right? Edible portion of mango is mesocarp. And mango is a fruit which has one of the highest vitamin A content. Okay, so that is 4,800 international units. And in terms of vitamin A content, papaya follows mango. Okay. So propagation type in case of mango is inarching, epicotyle grafting, and veneer grafting. Next, important seasons, if you see. So mango fruits are available from the months of March to mid-August, and therefore we go for pruning after the fruiting, that is August to September. Since mango trees have a big strata, so therefore the spacing that is maintained is 10 meters into 10 meters. However, we can also plant mango in reduced spacing, that is high density planting. And in high density planting, we go for spacing of 2.5 meters into 2.5 meters. The most important variety in high density planting is Amrapali. Then if you see here, I've mentioned polyembryonic rootstocks, Bappakai, Olor, Chandrakaran, and Goa. Important point to note is that polyembryonic varieties of mango usually are the South Indian varieties, whereas the North Indian varieties of mango are the monoembryonic varieties. And one of the problems in case of monoembryonic varieties or North Indian varieties is what? That is, they are alternate bearers, right? Okay. Then you see here some of the important varieties of mango. So the famous varieties and also for export varieties are Alfonso, Dasheri, Kesar, Banganapalli. Now, my first question for the day is, what is the problem in case of Alfonso fruits? Okay, what is the problem in case of Alfonso fruits? So you need to answer this question in the comment section. Important North Indian varieties are Lengra, Chausa and Dasheri, and Langra is a late variety. Mutant variety is Rosica, and seedless variety is Sindhu. Then if you see off-season varieties, Neelam, Niranjan and Madhulika. In the off-season varieties, if you see, the fruits are available from the months of January and February. Okay, so that you have to remember. Normal fruits are available in the months of March to mid-August, whereas off-season fruits are available in the months of January and February. Next, some of the important hybrids. Sometimes in exam they ask about the parentage of the varieties, or sometimes they may also provide the parentage and ask you to write down the hybrid. So in that case, this becomes important. So first you see Ratna is a cross of Neelam and Alfonso. So Neelam we have seen is a off-season variety. Sindhu seedless variety is a progeny of Ratna and Alfonso, right? Next, if you see Amrapali variety, so Amrapali is a cross of Dasheri and Neelam. And how can you remember Dasheri and Neelam? Amrapali variety, how can you remember the parentage? That is DNA. Okay, Amrapali variety, you can remember by DNA. D, Dasheri, crossed with Neelam to form Amrapali. Opposite parentage of Amrapali is here, Mallika, that is Neelam cross Dasheri. So accordingly, Mallika you can remember as NDM. Neelam crossed Dasheri gives Mallika. Again, similarly, you see Arka Punit. The parentage is Alfonso cross Banganapalli, and opposite is Arka Aruna, that is Banganapalli cross Alfonso, right? So you can remember as what? APAB. Arka Punit, Alfonso cross Banganapalli. And when you remember Arka Punit, automatically you can remember Arka Aruna because it is just the opposite, Banganapalli cross Alfonso. So the question for today, first question, if the spacing for mango cultivation is maintained at 10 meters into 10 meters, then what is the plant population in 1 hectare of land? Okay. So first question I've asked is what is the problem in Alfonso fruit? And this is the second question. That is, what will be the plant population in 1 hectare of land if spacing is maintained at 10 meters into 10 meters? Okay. So next crop we're going to see about is banana. So botanical name, genus is what? Musa. And there are two prominent species of banana. That is Musa acuminata and Musa bulbiciana. The common cultivated banana is Musa paradisica, right? Which are the hybrids of the crosses of acuminata and bulbiciana. So that also you can remember. And Musa paradisica is a triploid, right? So the common cultivated varieties of banana which we consume, it is a triploid, okay? That is why it explains what the seedlessness in case of banana. So origin you see, Musa acuminata is of Malaysia and Musa bulbiciana of Burma. What was the origin of mango? Indo-Burma region. So you can remember, mango, Indo-Burma region was the origin, and Musa bulbiciana is also Burma, right? Okay. Then the basic chromosome number of banana is n is equal to 11. Accordingly, diploid hoga to 22. So that way, okay. And the cultivated banana is triploid. I already told you, Musa paradisica. Inflorescence of banana is spadix, right? And in the spadix, there are 3 types of flowers: male flower, female flower, and hermaphrodite flower. So female and hermaphrodite flower matures first, okay? And you can see the fruitlets, right? In case of banana tree, the spadix, the fruitlets come out. Then later on, the male flowers come. And we remove the male flowers, right? You know that. Why do we remove the male flowers? So that more energy is diverted into the development of fruits, right? In the later part, we'll see that what is that removal of male flowers in banana called as. Okay, fruit type in banana is berry, and banana is a climacteric fruit. Banana fruits are said by parthenocarpy, right? Therefore, we do not need the male flowers, okay? Rhizome is a modified stem, and the plant type of banana is tropical, perennial, and herbaceous plant. Next, if you see, did you know banana pseudostem is actually a modification of leaf sheaths? It is formed by the tightly packed overlapping leaf sheaths. So this one you see, These are the leaf sheaths. Okay. For banana, the soil pH that is important is in the near neutral range, 6.5 to 7.5. Then if you see, so what is the water requirement in case of banana? 1800 to 2200 millimeter per plant per year. And banana is a heavy feeder of fertilizers. Okay. It's an exhaustive crop. So banana requires a high amount of potassium fertilizers. Okay. Then next, if you see, the important part is seasons of planting. So different types of land and different time at which the banana is planted. So wetlands, February to April is the planting time. For garden lands, it is November to February. And for hill banana, April to May and June, August is the time for planting. Commercial method of propagation in banana is sword suckers. And nowadays, tissue culture banana is also very commonly used for planting. So what is the advantage of tissue cultured banana? That is, those banana plantlets or the suckers are free from virus, right? Okay. So common method of tissue culture that is used is shoot tip culture. Then the spacing is 2.1 meters into 2.1 meters, and for dwarf varieties, we go for 1.8 meters in both ways and square method of planting. Then if you see the dessert varieties, that is the table varieties of banana, dwarf Cavendish, Robusta, Monthan, Rostali, Puvan, Nindran, Grand Name, Karpuravalli, Champa, Bhimkol, Biruvakshi, etc. And the famous varieties which are used for making chips is Nendran and Chipsona. You need to remember Nendran because it is a typical dual purpose variety. That means it can also be used as dessert purpose and culinary or planting purpose. Okay. Then culinary banana varieties are Monthan, Ashmonthan, Chakia. Tissue cultured banana variety is your grand name. So usually the tissue cultured plants that we get, um, those belongs to the variety grand name. Okay. For export market, Cavendish groups are generally preferred, and Hind banana examples are Virupakshi, Sirumalai, Namarai, red banana, Manoranjitham. Then if you see, these are some of the important cultural practices in case of banana, and these are, you see, typical terms, right? And you know that in agri-related exams, terminology-based questions are very, very common, right? So therefore, this four terms becomes very important for you. First is metalking. So we know that in banana, ratoon cropping is practiced, right? So at the time of bunch appearance, okay, one healthy sword sucker, okay, is selected as a daughter sucker. And rest of the suckers from that time, it is removed. Okay. Now, once the bunch is harvested from the mother plant, then the pseudostem is cut, leaving 60 centimeter from the ground. Okay. And this pseudostem stump will nourish the daughter sucker that we selected for our ratoon crop. Okay. Till the pseudostem withers and dries up. Okay. So this whole process is known as what? Metalking, the leaving of the pseudostem for nourishing the daughter sucker. So this process or this phenomena is known as metalking. Clear? Next, we'll go for propping. So we know that banana is a what herbaceous plant, right? And the pseudostem is a modified leaf sheath. Okay, if you've seen banana, it's not very sturdy, isn't it? So during bunch development, what happens is that the banana tree may topple down, okay, may fall. So in order to prevent that, we provide support to the pseudostem, okay, using bamboo or something like that. So that is known as what? Propping. Okay, the practice of providing support to the pseudostem during bunch development is known as propping. The next is your denaveling. So I told you that in banana inflorescence, that is spadix, we have three types of flower: female, hermaphrodite, and male. So female and hermaphrodite flowers will open first, will mature first, will develop into fruitlets, and later on what happens? The male flowers open. Okay, and you must have seen that in case of banana bunch, the fruitlets are there. Then after the female phase stops flowering, I mean is over, then uh, the flower, the male buds, once they mature, they just fall off, isn't it? They do not result in any fruitlets. So what happens is that it will only divert energy, isn't it? So what we do is that we remove that flower bud so that all the energy is now concentrated into what? Developing of the fruits. So that process of removal of the male flower bud after completion of the female flowering stage is known as denabling. Then we go for desuckering. So desuckering, as I've already told, that during the bunch, once the bunch is developing, okay, so there we select a very healthy daughter sucker, sword sucker, okay, and rest of the suckers are being removed. Even before bunching also, whatever suckers come out, they were removed. Only the sucker which arises during bunching, okay, so that is selected as the daughter sucker for the next ratoon. So this practice of removing unwanted suckers time to time in case of banana plants is known as desuckering, okay. So metocking will leave the pseudostem stump for nourishing the daughter sucker. Propping is the support for the pseudostem, right, that is provided during bunch development. And deleveling is the removal of the male flower bud, and desuckering is the removing of the unwanted suckers time to time in the banana plant. So these four terminologies are very, very important. Please remember them. Okay. Now the next question that is upon banana, you see, what should be the approximate weight of sword suckers to be used for planting material? I already told you that sword suckers are used for commercial planting of banana, right? So what should be the approximate weight of sword suckers? Next crop is a papaya. So, you know, the papaya botanical name is carica papaya. And a lot of things will see this suffix carica, right? Like the family, caricaceae, the pigment, carica xanthin, right? So in that way, you can remember. So origin of papaya is tropical America, and papaya is a cross-pollinated crop. You know that papaya is a what? Polygamous plant. Polygamous plant means it has different sex forms, dioecious, gynodioecious, right? So accordingly, since it has different sex forms, automatically it becomes what? Cross-pollinated crop, right? Okay. Apart from the fruit, you know, another very important product that is obtained from papaya is what? Pepin, isn't it? It is the latex that is obtained from immature fruits. Immature fruits as in 70 to 90 days old fruit, okay? They are usually commercially used for pepin extraction. Okay, you can see in the picture that they would score the immature fruits and collect pepin. This pepin is then dried and processed as a commercial product. Now, the enzyme that is present in pepin is pepsin, right? Pepin is used for what? As a meat tenderizer. So, why is pepin used as meat tenderizer? Because pepin has the enzyme pepsin, which helps in meat tenderizing. Okay, so that can also be a probable question. Propagation of papaya seeds. And you know that this is the only method of propagation that is being followed in papaya. Then important point is regarding the temperature. You see, papaya is a tropical crop, so it requires a temperature range of 20 to 35 degrees centigrade. But if the temperature goes beyond 35 degrees centigrade, then it leads to female sterility, right? So 20 to 33 degrees centigrade is very suitable. Up to 35 degrees centigrade, it's okay, but beyond 35 centimeters, centigrade, female sterility occurs. Okay. So next we'll see that the spacing, normal spacing is 1.8 meters in both the sides, row to row and plant to plant. That is in a square system of planting. But we can also plant papaya in high density planting. That time the spacing is 1.2 meters into 1.2 meters. Then growth regulators, if you see, ethyl at the rate of 200 ppm, GA at the rate of 50 ppm. So why do we use growth regulators in papaya? Because these growth regulators promote femaleness in the papaya plants. Okay, so if we'll have female flowers, we'll have more yield. So that is why these growth regulators are used. Okay, ethyl at the rate 200 ppm, GA3 at the rate 50 ppm. Even phosphon D is also used, but these two are the most common. Okay. Then you see, I have told you that it is a polygamous plant, isn't it? Cross-pollination occurs. So if it is a dioecious plant. Okay, if we plant in 1 hectare area, if we plant dioecious variety, then what should be the male and female plant proportion that needs to be maintained? So, for every 10 female plants, we would need one male plant as the pollinizer. Okay, if we grow dioecious variety, then we would need one pollinizer male plant for every 10 female plants to have satisfactory fruit set. Okay, all right. This will not happen in case of gynodiaceous variety, right? Okay. Then next we'll come to the varieties. So gynodiaceous varieties, Pusa majesty, Pusa delicious, Sunrise solo, solo, Cool honeydew. And dioecious varieties are Pusa giant, Pusa dwarf, Pusa nadha, CO5 and CO2. So I told you this is the thing that dioecious varieties hoga to kya hoga? We have to maintain a plant population of 1:10 male and female plants, right? Why? For pollination. लेकिन ये problem किसमें नहीं देखेंगे हम? In case of gynodiaceous varieties, right? Okay. Then transgenic varieties, Sunup and Rainbow. Sunup is the first transgenic variety, लेकिन commercial propagation के लिए Rainbow is the first variety. ठीक है? Transgenic. फिर highest papain extension. Okay, for that we use CO5. चलो. The third question for the day is, which variety of papaya is most suitable for high density planting? So we've seen the spacing for high density planting is 1.2 meters both sides in case of square method of planting. So which variety of papaya is most suited for high density planting? Okay. So these questions that I've asked in this session, please answer them in the comment section. That's all for today. And stay tuned to our channel for more such agri-related content. Thank you.